topic today is a really exciting area of research with a lot of new research kind of coming out in the last several years. And our speaker today is Omega-Quant founder, Dr. Bill Harris. Dr. Harris has a PhD in nutritional biochemistry and has been studying omega-3 fatty acids for the past 45 years. As of February 2024, he has 345 omega-3 related publications and has been the recipient of 8 NIH grants on omega-3 fatty acids. In 2004, he co-developed an omega-3 blood test called the Omega-3 Index. And in 2009, he formed Omega-Quant Analytics to offer the test to researchers, clinicians, and consumers. In 2020, he started the Fatty Acid Research Institute to accelerate the discovery of fatty acid and health relationships. And his scientific productivity was recently ranked among the top 2% in a survey of scientists worldwide, which is really amazing. And today he will be discussing omega-3s, the omega-3 index, and their relationship to atrial fibrillation, or A-Fib. So that is all I have to say, and I will turn it over to you, Dr. Harris.
Okay. Thank you, Kelly. And hopefully everybody can see that. You'll let me know if they can't. Great. Okay, so we're going to talk about omega-3 and A-Fib. What's the real story? Well, I wish I had the real answer. I've got a lot of information that's been accumulating recently, but the real answer is something I think we're still waiting for. Let's see if I can get this to advance. Okay, that maybe advances it. Here we go. So here's the way we're going to do this. I'm going to start by, of course, definitions and prevalence of A-Fib, who's at risk, some of the risk factors, and the medical relevance of A-Fib. And we'll do that right here. Here's a nice little graphic from the CDC showing that what A-Fib is. It's the most common type of heart arrhythmia. On the left, you'll see what a normal heartbeat looks like. The atria, of course, are the two upper chambers, and the ventricles are the lower chambers. And it's supposed to be on the left, the atria are supposed to contract at the same time and push the blood down into the ventricles. But in A-Fib on the right, the timing of the opening of the into the ventricles is random, and it's not coordinated, and so it makes the heart beat less efficiently. And so that's the problem with A-Fib. That's what it is. Many people, not just some, many people who have A-Fib don't know it because they don't have any symptoms. Others may experience one of the following: irregular heartbeat that you actually feel, heart palpitations, which would be, rapid fluttering, pounding, lightheadedness, extreme fatigue, shortness of breath, chest pain. All these things can be part of A-Fib. So it's not an irrelevant issue. This is old data, but the point is still made here. If you on the left-hand side is the U.S. population and the age of people in the U.S. population. As you see, older we get, there's fewer of us around. But the people who have A-Fib are almost all up in this, over 60 category. So it's really, the prevalence is 5 to 8% in people over age 65, I believe it is. I've got some things covering up here. Right, so it's really an age related, very, very rare in most, there we go. Here is a information rich, too rich slide, but I wanted to point out up in the upper left-hand corner, roughly 2010 and 2020, 2.7 million people in the U.S. had A-Fib, about twice as many in Europe. 2% of the people under age, 2% of the A-Fib is in people under 65, 9% is of the people over 65 have A-Fib. So it's quite common. Risk factors kind of here in the central area, that some of the common risk factors for most diseases, particularly cardiovascular diseases. I would want to point out sleep disordered breathing, like sleep apnea, is definitely a risk factor for it too. So trying to control all of these things will help reduce risk for A-Fib, but it's a big problem. It's been around for a long time. So that's a short intro on A-Fib. Let's get to the omega-3 part of it. So I'll give you a little background with some one animal experiment. There have been many, some epidemiology, some short-term clinical studies, and major omega-3 trials used to prevent A-Fib. Basically what I'm going to say is between 2000 or maybe 1995 and 2010, there was great interest in trying to control or treat A-Fib with omega-3. So that the expectation has always been that there's a benefit of omega-3 and A-Fib, and which is what was so surprising with some of the more recent trials showing an increased risk in A-Fib. We'll look at some epidemiologic studies that show decreased risk. We'll talk briefly about potential mechanisms of action and some official recommendations. So one study in animals, and this was done in rabbits, and this is the idea of giving rabbits, in this case, omega-3, and then doing tests to force the heart to go into A-Fib. And if comparing control or sunflower oil or fish oil, what they saw back here in the early 2000s was that there was less incidence of A-Fib. The heart was less able or likely to go into A-Fib when the animals were on fish oil. And so this, of course, made us assume that the higher omega-3 would be good for A-Fib. I'm not going to go over all these studies, but past observational studies that compared the risk for A-Fib as a function of either omega-3 or fish intake, or even circulating levels of omega-3, found only neutral or favorable relationships, never an adverse relationship. So this was, again, the expectation. These papers were published between 2009 and 2022. Here is one study. I'll point out this was from the cardiovascular health study, and this one looked at the association between eating oily fish, tuna or other baked or broiled fish, and the intake of fried fish or fish sandwiches, and how they relate to the incidence of A-Fib in this one study. 5,000 people in this study over age 65. So it's a very rich population for A-Fib. 980 people developed A-Fib over 12 years. And what they found was, if you look here as this is the low intake of oily fish, this is high intake of oily fish. As the intake of oily fish increased, the risk for A-Fib, this is the worst case scenario, the risk for A-Fib was the lowest. The more people ate trans fat rich foods, we'll just say what it is, fried fish, it increased the risk for A-Fib. So here again, consumption of tuna or other boiled non-fat fish or high fat fish, but not fried fish or sandwiches, associated with a lower incidence of A-Fib. Hence the expectation, as always said. Another study from the early 2000s. We're looking here and trying to prevent atrial fibrillation post-CABG, post open heart surgery. This is a very common side effect or outcome, short-term outcome of CABG or open heart surgery is developing A-Fib. So here we had there were 160 patients awaiting CABG surgery. They were randomized to get EPA and DHA for 5 days before surgery and then throughout. And then the endpoint in this study was the development of A-Fib over the next certain level of A-Fib over the next few days. And what they found here was evident from the graph. The people who got omega-3 were much less likely to develop A-Fib. Who got omega-3 were less likely to develop A-Fib over that time. Half as many people developed actually A-Fib post-CABG on omega-3, and that was very encouraging. We thought this could be something that could be used in surgery to prevent development of A-Fib. In another study, again, get 2 groups of people with A-Fib who underwent cardioversion, which is a in-hospital or in-clinic treatment that electrically shocks the heart and brings it back into normal rhythm. Theoretically, that's what it's supposed to do. And the question was, could having omega-3s on board help retain that sinus rhythm after you've gone through cardioversion? And in this case, I'm looking out for a full year, they found an increased ability to retain normal sinus rhythm in the people that got omega-3 versus placebo. Again, very positive finding. Unfortunately, the study I just showed you is this one right here, where the omega-3 group had less developable, excuse me, who were able to maintain sinus rhythm the longest compared to placebo group. But then there have been several other studies since, and virtually none of them really found the same effect as this study found. So we're beginning to see a differential finding in terms of omega-3 and A-Fib, as far as how well it protects you, allows you to stay in sinus rhythm after cardioversion. All this led to a major clinical trial published in JAMA in 2010. This was a study where the omega-3s were being used, to treat A-Fib. Okay. And the title of Efficacy and Safety of Prescription Omega-3 for the Prevention of Recurrent Symptomatic A-Fib, randomized trial. There were, randomly assigned to placebo, and they got 8 grams a day of omega-3 for the first 7 days after they came in. With these people all had symptomatic A-Fib. Either it was called paroxysmal A-Fib, which is infrequent, comes and goes, or persistent A-Fib, which is there all the time. Again, large dose, 8 grams a day for the first 7 days, and then they went on 4 grams a day for the next 24 weeks, so roughly a half a year study. And then again, they were looking at the primary endpoint of developing symptomatic A-Fib. And what did they find? Here are the curves, event-free survival curves. Basically, they're no different. You go out to the 168 days, there was no difference between the omega-3 and the placebo in the effect on preventing the recurrence of A-Fib. So this was a disappointing finding, but you get what you get. And it certainly caused people to believe that there's no reason to give omega-3 to prevent A-Fib. The next study was a little more focused. This is called the OPERA study. And here we're not looking at people with A-Fib at the beginning. We're looking at people that are undergoing cardiac surgery. Again, I mentioned that one of the side effects of cardiac surgery, open heart surgery, is A-Fib. Here was 1,500 patients in really a multi-international study, Italy, Argentina, and the U.S. They were randomized to either olive oil placebo or supplements, omega-3 supplements containing 8 to 10 grams of omega-3 in form of Omicor, which is Loveza. They were given preoperative loads of 10 grams of omega-3 for 3 to 5 days, or 8 grams over 2 days beforehand. And then they were on 2 grams a day after surgery until post-op day 10. And again, the outcome was, did they develop A-Fib during or in the post-op phase? Hypothesis was that there would be a reduction in risk. What they find? Again, no effect. After all this work, the authors were disappointed to find that there was no benefit and no reduced risk for A-Fib post-op, post-cardiac surgery. So again, no increased risk, no damage, but there was no benefit. So despite these early studies in animals and small trials in humans treating A-Fib with omega-3, the larger studies really haven't found an effect. So the next question is, what about studies where they're not targeting A-Fib as a primary outcome, but they're looking at A-Fib as potential adverse effect in the study? And that's where it all hit the fan a couple of years ago. And this is a meta-analysis summary of recent randomized trials. I have one in a box called Reduce It. Reduce It was a study with pure EPA ethyl esters, 4 grams a day. This is the first study that found an effect of higher risk for developing A-Fib in the omega-3 group compared to the placebo group. And they found that this is a odds ratio here, 1.35 or 35% increased risk and we'll look at that in more detail in a moment, for A-Fib. And once this study found it, other studies that had been done looked back at their data and started to see what they found. And if we look at low-dose omega-3 studies, here's 4 of them: Vital, Ascend, Risk and Prevention, and Risk and GC Heart Failure. There was a small increased risk for A-Fib in really none of them significantly, but when you put them all together, there was about a 12% increased risk. If you look at the high-dose studies recently, all done in patients at risk for cardiovascular disease, you can see a higher increased risk. If you put it all together, the overall was about 25% increased risk for A-Fib across these 8 studies. So to summarize it, if we look at the low-dose studies, the rate of A-Fib in the placebo group was 3.7% of the people on placebo developed A-Fib, and 4.2% developed A-Fib on omega-3. High-dose studies, 2.4% versus 3.5%. If you add them all together, through about 3.3 versus 4%, a 21% relative increase in risk, but about 0.7% absolute risk. Excuse me, absolute increase in risk. And as you can see, it's sort of dose dependent. There was very little effect, but not nothing in the low dose studies, but more of an effect in the high dose studies. There is a term we call number needed to treat when you're looking for benefit. There's also the number needed to harm, which is applied in this case. It was 200, which meant that for every 200 patients receiving omega-3, one person on average would acquire AF. So put that in there just for your perspective. Small increase, but not nothing. Because of these studies, and particularly these meta-analyses, both the package insert for Vascepa and the package insert for Loveza now mention that there are possible concerns with developing A-Fib or A-flutter. They just basically say to watch for it. It's a possible association, and perhaps people should be monitored more carefully if they're on omega-3 for the A-Fib outcome. Now, what about those are randomized trials? What about some observational studies? And there's some very compelling observational data. The first one I'll talk about here is from Frank Chin and the FORCE collaboration. And these studies are looking at the question, is there a relationship between blood levels of omega-3 and risk for A-Fib in very large, long studies, not just short-term studies with acute supplementation. And so this study pooled data together from 17 prospective cohorts, about 55,000 participants, all free of A-Fib at the beginning of the study. And again, these are cohorts like the Framingham study or the Women's Health Initiative study or EPIC or a variety of others where healthy people typically are recruited at the beginning and then tested for everything under the sun, and then they are followed for many years for the development of different diseases. And all these 17 studies had fatty acid levels measured in the blood before the study started. And then they were followed for, again, about 55,000 people over about 13 years on average of follow-up, much longer than any randomized trial goes. There were 7,700 cases of atrial fibrillation. So the question was there a relationship between the omega-3 level at baseline and who developed A-Fib? And what Frank and his colleagues found was that if you look at the relative risk of incident A-Fib, comparing the top to the bottom quintile of omega-3 levels in the blood. So we're looking at people with the highest omega-3 level versus the lowest. And when you do that comparison, you see, if we look just at plasma level, or excuse me, circulating levels of EPA-DHA, there's roughly a 12% lower risk, statistically significant, lower risk of developing A-Fib if you had the highest omega-3 levels versus the lowest. Now, again, this is not a supplementation study. This is just typically omega-3 levels, which are driven by different intake of omega-3 fatty acids. There may be some genetic factors that may raise or lower omega-3 levels, but it's not a supplementation study. So in this case, higher omega-3 was beneficial. And the authors finished the paper by saying biomarkers of omega-3 fatty acids, DPA, DHA, EPA, and DHA demonstrated inverse association with incident A-Fib. In the absence of randomized trials examining the long-term dietary omega-3 intake and risk for A-Fib, our results do not suggest that higher levels of these fatty acids are associated with harm. So that's going the other way, higher omega-3 beneficial effect. This was also seen in a very recent study from what's called the Million Veterans Study. There weren't 1 million people in this study, but there were 301,000 people, veterans who completed this is food frequency questionnaire. So this is not based on blood levels. This is based on reported intake. And the reported intake roughly around 200 milligrams of EPA and DHA. The mean age, 65, mostly men, mostly white. But in this figure, they're showing the risk of developing atrial fibrillation over the dose of omega-3 that people reportedly were eating. And you can see that the higher the omega-3 dose chronically in the diet, the lower the risk of atrial fibrillation. So again, dietary levels, even at pretty high dietary levels, there's really no increased risk. There actually is a decreased risk. And the authors said at the end, contrary to our hypothesis, dietary omega-3 was not associated with the higher risk for A-Fib, but was inversely related to A-Fib risk in a nonlinear, meaning it went down and then was flat. And so again, beneficial. Just to add confusion to the story, here is a recent study from the UK Biobank, which is a very large cohort of individuals in the UK that have been followed for now 10 years, about 470,000 people at the beginning of this study. And they were asked, as one of the many questions, do you use fish oil supplements? Yes or no? And there were about 30% that said they used fish oil and 70% did not. And so the question was, well, over the ensuing 10 years, who developed A-Fib? And this was all part of the medical records. And what they found was the people who said they were fish oil users had 6.5%, 6.2% developed A-Fib, and 5.2% who were non-users developed it. And this was statistically significant, about a 10% increased risk. You can see the difference here on this curve. So here's a study, got to deal with the data that doesn't show benefit, but shows a slight increase in risk for people who say, now we don't know the blood levels in this particular study have not been reported yet. So whether maybe they have, I think we did actually do that. I think that's coming. Hold on a second. I think I did. I'll look at that in a minute. We'll get to that in a second. In any event, omega-3 treatment trials in patients at risk for cardiovascular disease did show an increased risk for A-Fib, whereas most epidemiologic studies, most, not all, have found that higher intakes or higher blood levels of omega-3 are associated with lower risk for A-Fib. So we'll get to some mechanisms later, but that is the lay of the land at the moment. The question, what about stroke? Well, why would we ask that? Well, one of the main risk factors for stroke, ischemic stroke, blood clot related stroke, is having A-Fib, which is why people who have active A-Fib are typically put on blood thinners so they don't develop blood clots that go to the brain and cause a stroke. So the biggest concern about A-Fib is not A-Fib itself so much as the increased risk for stroke. So the question is, do omega-3s increase risk for stroke? Well, let's look at that. Here's a recent paper from it's part of the FORCE Consortium, led by Dr. O'Keefe in Kansas City. We looked at, in this study, the outcome was, of course, incident stroke. We had 29 international prospective cohorts, blood level measured of omega-3. 183,000 people were involved in these 29 studies. With about 14 years of follow-up, there were over 10,000 strokes occurred among all these people. 8,000 of them were ischemic stroke, meaning blood clot related strokes, and about 1,000 were hemorrhagic, meaning not blood clots, but blood, a brain bleed. And so we were looking in this study at the relationship between omega-3 levels in the blood and risk for stroke. And what Dr. O'Keefe and colleagues found was shown pretty clearly here on this graph on the right. If we look at the increasing levels of omega-3, and this is for total stroke over here, comparing the people who had the lowest omega-3 levels, here is the reference group, to those who had the highest omega-3, we saw that the higher the omega-3 level, the lower and lower the risk for stroke. And this was driven by ischemic strokes, blood clot-based strokes. There was no relationship with hemorrhagic strokes, meaning there was no increased risk for bleeding, a bleed caused by a stroke. And if we compare really the highest to the lowest level, there was about a 17% lower risk for stroke in people that had the highest omega-3 levels. So that's very comforting, knowing that one of the sequelae of A-Fib can be a stroke. Finding lower risk for stroke with omega-3 is a good finding. And I mentioned also earlier, I talked about the UK Biobank study in fish oil users and fish oil non-users, and there was a slightly increased risk for stroke. As part of the O'Keefe meta-analysis, the UK Biobank was included, and we do have blood samples on a subset of people in that study. And we looked at blood DHA levels as the biomarker, primary biomarker, the DHA level in the UK Biobank. Comparing the highest to the lowest level, there was actually a lower risk for stroke in the UK Biobank. When you measure blood levels, there was, that is opposite of what was reported for people who said they were taking fish oil supplements versus those who were not. I tend to believe a biomarker more than a reported use of fish oil. So it looks like a beneficial effect in stroke. This is not new. Actually, the first study that pointed out the increased risk for A-Fib was, as I mentioned, the REDUCE-IT study, where they gave 4 grams of EPA ethyl esters to high-risk cardiovascular patients, and they found increased risk for A-Fib. But in the same study, they also looked at fatal or non-fatal stroke, and they found a 28% reduction in risk for stroke in the people that got the omega-3. So again, higher omega-3 is associated with lower risk for stroke, regardless of whether it might induce an increase of A-Fib or not. The findings in REDUCE-IT were not potentially surprising, because the study on which REDUCE-IT was based, which is called JALIS, this is a study in almost about 19,000 Japanese men and women from the early part of the century, published in 2007. Where they looked at the effects of EPA on cardiovascular disease, and they also looked at stroke. And in this study, they broke it down into people who had no history of stroke and the people who had a history of stroke. And of course, most people in the study did not have a history of stroke, and there was no difference, EPA or no EPA, on the risk for stroke in people who had not had a prior stroke, so there was no increased risk. But interestingly, in the people with a history of stroke, the people who got EPA had a lower risk of developing a second stroke. Again, very comforting, a good relationship between omega-3 and risk for stroke. Well, let me get to some possible mechanisms of action. And this is still way up in the air. I don't think we don't know the whole story yet. I wish we did. First one we'll look at is statistical problems that perhaps overestimated the increased risk of stroke, which is kind of fascinating. There is certainly a hypothesis that increased omega-3 in cell membranes could alter membrane properties that would make the firing of the atria, the contraction of the atria, inappropriate contraction, more likely, which is A-Fib. Another hypothesis, particularly from Dr. O'Keefe and his colleagues, is that omega-3 could increase vagal tone, meaning the vagus nerve, this parasympathetic tone of the body, is stepped up by omega-3, and in some people that can predispose to A-Fib. So the first one about statistics. let's first of all just look at this table that I copied out of this paper from Circulation a couple of years ago. And these are all the studies that we just looked at early, the randomized trials in which omega-3 was given, and there was a overall increased risk for atrial fibrillation. And here is the incidence of A-Fib in each one of those studies, and in the omega-3 group. And you can see 3.6% increased, or excuse me, there was a 3.6% increase of about a 2% increase in these studies, about 5% in this study, 12% in this study. So that's the incidence of A-Fib, which is the outcome of interest. Here's the incidence of all-cause mortality, meaning who died of anything. And the percent of people that died was roughly about 3 or 4 times higher than the people who got A-Fib. This means, so here they have the ratio. What that means is explained up here in this first sentence. Simply put, if omega-3 fatty acids reduce the mortality rate or dilute delayed risk for death, then the omega-3 treated patients had more time, more person time, or more opportunity to actually develop A-Fib than the controls did, which would potentially inflate the apparent incidence of A-Fib in the omega-3 arm compared with controls, and could contribute to the observed outcome. Or put another way at the end, because the majority of studies have potentially substantial bias due to what's called informative censoring, meaning they didn't take into account the fact that people were living longer on the omega-3. The true risk for A-Fib from omega-3 supplements remains unknown. And so that's another perspective on this, that maybe there is a statistical fact going on here that makes it look like they're at higher risk, but maybe it's not that high. That's still an open question. Another potential mechanism of action is a membrane effect. Here's a rough idea of the risk for A-Fib on fish or fish oil intake. At low levels, there's increased risk. If you increase levels to medium levels, then you drop the risk, and then it looks like if you're taking pharmaceutical high doses of omega-3, it seems to go back. It's a classic U-shaped curve. And their hypothesis is, without going through all the details, is that there's certain receptors, stretch receptors in the atria, that if the omega-3s make the membrane less stiff, they might be more likely to be activated and cause a A-Fib kind of reaction. So this is a more of a theoretical mechanism than anything else, but it certainly kind of explains this U-shaped curve, that too little is not good, maybe too much is not good. And there's a Goldilocks zone here in the middle where there's just the right amount. And finally, I mentioned potential that increased vagal tone could be something that would be causing this. We do know that omega-3 enhances vagal tone. And the evidence for this is it reduces resting heart rate. Omega-3s augment heart rate variability. They make heart rate variability, which is a good thing, go up. And they improve post-exercise heart rate recovery. All these are signs that the omega-3 treatment is basically stimulating the parasympathetic side of the heart, carried by the vagal nerve. And then the authors of a paper Dr. O'Keefe has written here recently, the omega-3 dose-dependent amplification of vagal tone might be particularly problematic for people at risk for bradycardia. So this is slow heart rate, bradycardia-dependent A-Fib. And there are people like that who have a very slow heart rate, and they develop A-Fib because of that. Such individuals who might be in this category are people who engage in large amounts of very strenuous exercise that can actually increase risk for A-Fib, who have untreated sleep apnea, or have what's called sick sinus syndrome, chronically slow heart rates. And so these are all hypotheses that need to be tested. What's disappointing to me is that the people who have run for example, the Reduce It study have not yet published a paper identifying what are the characteristics of those people on the omega-3 group who developed A-Fib compared to those who did not develop A-Fib. In other words, what were the risk factors or the factors that were associated with developing A-Fib And that would be very helpful for us to understand more about why certain people develop AFib and certain people don't. Again, remember we're talking about maybe a 1% absolute increased risk, which is not much. But it is real. And I think we'll end with this one, consumption of omega-3. This is a quote from, again, Dr. O'Keefe's recent manuscript, and I agree with this. Consumption of marine sources of omega-3, such as fish, seafood, has been consistently associated with lower risk for developing AFib. And the ideal dose appears to be somewhere around the 600 to 750 milligrams a day, if we can base it off of blood levels. In contrast, higher dose omega-3 interventions over 1,800 milligrams a day, which is the intervention studies, appear to increase risk for AFib, although the absolute risk is small, about 1%. And if we want guidelines, I think this makes sense. Individuals with a history of AFib or risk factors for vaguely mediated AFib, like a very slow heart rate, should be advised to avoid pharmaceutical treatment with omega-3 and instead consume at least 2 servings a week of non-fried fish, seafood, as recommended by the American Heart. And I think that does make some sense. So that is the story as we have it today. Happy to, I wish I could say that it's crystal clear. This is exactly what it all means, but we're in a developing field and there's more research needed here. Back to you, Kelly.
That was great. Thank you so much, Dr. Harris. We have got a lot of questions, so I'm going to kind of start at the top and go through, and we're going to answer as many as we can. And then again, like I said at the beginning, if anyone has a question and that we don't get to, please feel free to email it to me at kelly@omegaquant.com. All right, so I'm going to start at the top. Does the ratio of EPA and DHA make a difference for AFib? I'm assuming they're talking about in supplementation.
I assume we don't know that. We do know that in 2 of these pharmaceutical studies, well, 3 of them. One of them was EPA alone, reduce it, and the other 2 were EPA plus DHA. And in both cases, all 3 cases, there was an increased risk. So it apparently does not make a difference that it's EPA alone versus EPA plus. Nobody's done a study with just DHA. So we really don't know. But it appears to be both.
Okay. Do you know of any genetic markers which could affect the risk of AFib with relation to omega-3 intakes and potentially account for the differing results?
The short answer, no. I know that there are what they call, prospective or risk scores based on genetics for people who are at risk for AFib. I'm not real familiar with them. I just know they've been adjusted for in some studies. So theoretically, that makes a lot of sense. You could, if there was a genetic panel that would identify people at high risk because of genetics, those people might be advised to not take omega-3. Although I'd love to see a study where you randomize those people to omega-3 and see if it made a difference.
Do omega-3 supplements cause an increased risk of bleeding? I thought this was debunked, but it could be wrong.
It has been debunked. In fact, in the one study we showed here called the OPERA study, where they took people who were having open heart surgery and gave them a bunch of omega-3 beforehand to try to prevent AFib, one of the things they discovered was there was actually less bleeding, less need for transfusion in the people that got the omega-3 compared to the placebo group. And there's been many studies that have shown no increased risk for bleeding. So it's an urban legend that I wish would die, but doesn't seem to want to.
Yep. Is it possible that DPA is more effective than both EPA and DHA in reducing the risk of stroke?
No real evidence for that, but anything is possible. Again, that's would have to be tested. We don't eat very much DPA. The DPA we have in our blood is pretty much synthesized from EPA, and it's sort of appears to be a reservoir for maybe the production of DHA, but DPA levels don't vary according to risk for heart disease, particularly. Or actually, you look at Japanese versus Americans, you look at their blood. The Japanese have higher levels of EPA and DHA than Americans by far, but the DPA levels are the same in Japan and America. So I don't think it's helping much.
Do we know if the people in the Reduce It trial, the ones who developed A-Fib, were they put on blood thinners?
No, not necessarily. There were no more people on blood thinners in the active arm, in the omega-3 arm, than the placebo arm. I mean, were they eventually after the study was over? Maybe. Yeah, we
Possibly.
don't know.
Yep. All right, this question's a little, can you explain the difference between Vascepa versus supplementing with EPA and DHA based on the Omega-Quant report in people diagnosed with A-Fib? So I think they're asking if there's a difference between Vascepa and the a combo EPA DHA supplement.
Right. And if people just taking Vascepa, which is just pure EPA, would have a higher EPA level on the omega-3 report.
Right.
And the DHA level would probably be no different from normal. People taking EPA and DHA naturally have higher levels of both EPA and DHA, which I think is the better way to go. Now, how all that relates to A-Fib, we really found in our A-Fib study with the FORCE group that blood levels of DHA and blood levels of EPA plus DHA and DPA were all associated with better outcomes. EPA levels per se tended to be higher in people that had less risk, but it wasn't statistically significant. So the action seemed to be, in terms of blood levels, was the longer chain omega-3s, the DHA particularly.
In the Reduce It trial, what fish oil product were they using?
Yeah, Vascepa, EPA ethyl esters, 4 grams a day.
All right, we've got some more that have come in here. Oh my goodness. Does absorption of omega-3 improve if taken with food? Yes.
Yes. You know that one.
Yeah. Any studies on people who exercise strenuously who take omega-3 and their risk of A-Fib? So does exercise modify any of that?
Yeah, that would be a great study. In fact, that's kind of what's part of what the implication of some of this data is, that that's a group of people that ought to be studied, given omega-3 or not, placebo, and see if there was an increased risk for A-Fib.
So several people have asked here, did any of the studies you discussed use the omega-3 index as a biomarker for the atrial fibrillation? So did any of the studies you went over use the omega-3 index?
Yeah. Certainly the FORCE study that looked at omega-3 levels, they didn't use just the omega-3 index. They used, in the FORCE consortium studies, we use whatever blood omega-3 biomarker any given study has. But when we correlate the levels reported in those studies with the omega-3 index, the highest level of omega-3 in that area is 7 to 8%. So it's approximately, if we had done omega-3 testing in omega-3 index testing in all of them, it would be around 7 to 8%. We still think that's the most optimal level for omega-3 levels for a variety of conditions: lower risk of stroke, lower risk of heart disease, lower risk of death, total mortality, lower risk of dementia. How it relates specifically to higher risk or to risk of A-Fib, we really don't know, because we didn't do all the analyses in all those studies.
And then the next two questions, last two on here are there any studies comparing the ethyl ester form of omega-3s with triglyceride form? Does that make any difference in this particular context?
Yeah, right. So there are 8 studies in this meta-analysis of randomized trials where A-Fib was increased. 7 of those 8 all used ethyl esters. One of them used a triglyceride. It's the OMIMI studies, the one that used the triglyceride. And they, of course, that was also done in very old people where A-Fib risk is higher. But anyway, there wasn't a clear distinction between the ethyl ester studies and the one that used triglycerides. So I don't think that's the deal.
Okay. All right, we still got some rolling in here. The next one, you mentioned possibly that Goldilocks zone, the too high and too low for omega-3 levels for increased A-Fib risk. Is there any evidence or do we have any idea of what level of would be too high on the omega-3 index? Do we know that yet?
It's very tough to say. From the epidemiologic data, the highest levels of omega-3 were around 8%, and that was protective. In the randomized trials done for short periods of time with high doses, 8% was probably where the omega-3 index was there too. Again, we didn't measure it and reduce it. But in strength, which was another one of the high dose studies, where they did see some increase in AFib, the highest omega-3 was probably in the 9% area. 4 grams a day of omega-3. So there isn't a cut point above which AFib risk really goes up based on the omega-3 index. Again, this is all still, I think, a developing story. My fallback position still is based on the totality of the evidence, is that a higher omega-3 up in the 8% level is good for many, many things. And there may be some individuals who, again, are susceptible, perhaps that heavy exercisers, that might be, they might not want to have an omega-3 index that high. It's always good to know what your index is.
And you can always eat fish.
You can always eat fish.
Always eat fish.
Yeah.
Did any study group the participants into their primary dietary macronutrient ratio type to determine if a certain macronutrient type, high carbs versus high protein versus high fat, influence AFib more than the amount of fish oil?
Interesting question. No, the answer is no, they did not. Most of those studies, randomized controlled trials, don't care much about diet, and they don't spend the money to quantify the diet in those people.
And the next one here, how can I correlate the recommendation of 600 to 750 milligrams per day to my own omega-3 index levels?
That, that was a back calculation from blood levels is where that's 600 to 750 came from in Dr. O'Keefe's paper. That probably correlates to an omega-3 index in the 6 or 7% area, I would think. I think that's not quite optimal. It's not bad. It's much better than most. But our experience usually is you need around 1,000 milligrams a day to get to the 8%, and none of those none of the high dose studies of the pharmaceutical products were that high. They were were that low. They were 1.8 grams or 4 grams. Or 2 grams versus 4 grams is what was there. None of them were taking roughly 1 gram, which we think is enough to get the omega-3 index up to about 8% in most people.
All right. Do you have any thoughts on how omega-6 or omega-9 fatty acids impact AFib/stroke risk? I know that's a whole different can of worms there. So
yeah, omega-6 is, when we've looked at omega-6 levels predicting cardiovascular disease or diabetes or total mortality, we find that higher levels of the primary omega-6 fatty acid, linoleic acid, higher levels are associated with better outcomes, lower risk for all of those things. So a high linoleic, I think it's been looked at in AFib and was not related to risk for AFib. Either that, or maybe that's a study that's going on right now, not published yet. But I think there's not been an increased risk for AFib with higher omega-6. It may be lower risk.
Okay. Does overprocessing of EPA in Visipa cause isomerization of the double bonds leading to AFib?
No. Okay. That's not the problem. you can take a Vascepa pill and analyze it in the lab, and it's not got isomerized double bonds in it. So that's
Okay.
not.
All right. All right. It looks like you've answered all the questions we've gotten so far. I will say we got a couple of questions in the chat that were more personal, individual questions. If you do have an individual question about your personal omega-3 situation, please feel free to email us at kelly@omegaquant.com, and we'll be happy to answer those kind of on a more personal level. If you also would like to make an appointment for a consultation to talk about your specific individual omega-3 levels of your omega-3 index test results that you've gotten done, you can do so on our website and make an appointment to speak to somebody. All right. Let's see. Is a person who takes up to 10 milligrams omega-3 as an anti-inflammatory for arthritis predisposing to A-Fib? Well, I think that's the question, right?
Yeah, and it probably is 10 grams, not 10
Yeah.
Don't know.
No.
It would worthy of investigation. Again, it had to be a pretty big study, but because again, we're only showing a 1% increase in risk. So to detect that, you need quite large studies to statistically detect a 1% change in risk. So I don't know. It's an open question.
All right. It's looking like we've got most of all of the questions. I think we're gonna wrap it up here. Thank you everybody so much for attending. Thank you, Dr. Harris, for this awesome presentation. This was really, really informative, and there's so much interesting research on this. Thank you, everybody. And like I said, you will all be receiving the recording of this webinar in your emails, along with the CEU certificate from CDR if you are eligible for those. Thank you for attending. And I think that's it for today. All right.
Bye-bye.
Bye, everybody.